In this video, we're going to talk about the first part of photosynthesis, the light reaction. Before we do that, we're going to talk about the parts of the cell involved in it, or the parts of the chloroplast involved in that, and then we're going to talk a little bit about the properties of light that are important in there. But you can see this guy is Melvin Calvin. He is credited with the Calvin cycle, which is where carbon is fixed into sugar. We're not going to do that in this video. We'll cover that in a later video. We're going to focus on this part where the light reaction occurs, where chlorophyll molecules absorb sunlight and they use water and they split the water and they release oxygen, and they really capture that energy in the first part of photosynthesis. So first, let's talk about the parts of the plant that are involved in that. Okay, so starting on the outside, right, the first thing we need on the outside is we need a way to get carbon dioxide in and oxygen out, right? So these are little gates called stomata. They can actually open and close. You can remember stomat, like this is like the mat, the front mat on the leaf that's allowing things in and out of the leaf. Okay, so this is kind of important for that first step. Then if you look inside that leaf, there is an area in the middle that's called the mesophyll. And the mesophyll is where the photosynthetic cells are. So the little green dots within the photosynthetic cells are called chloroplasts. So they're the organelles within the cells that are involved in photosynthesis. And if we zoom in on a chloroplast, we can see that it's got these little green stacks of membranes. They're called grana. You can remember GR for green. Okay, those membranes are called thylakoid membranes, okay, and that they're green because they're full of chlorophyll, okay, and then around them is this sort of clear area called the stroma. So the stroma is not involved in the light reaction, it's not absorbing sunlight, but it's involved in that second step where carbon is fixed, or carbon is converted from carbon dioxide to glucose, to an organic molecule. That's what we mean when we say carbon fixation. Okay, so let's talk a little bit about the properties of light. So in terms of all the light in the electromagnetic spectrum, most of it coming to us from the sun is in this very narrow range called visible light. And if we expand out visible light, we can see that going from shorter wavelengths to longer wavelengths, we have all these different colors, from violet to blue to green, yellow, orange, red. And then when you go off of red, you're into infrared, right? Those are the invisible wavelengths we can't see. But since most of the light from the sun is in the visible spectrum, plants are adapted and have evolved to be able to absorb visible light. But they don't absorb all visible light equally. So if we look at plants, plants are green. They're green because they are reflecting the green wavelengths of light. And the violets, the blues, the oranges and the reds are being absorbed by the plant. So that's how we can tell what types of light are most useful for plants and what are least useful, right? So the green is reflected. And about 100 years ago, a guy named Engelmann did a little experiment. He wanted to find out how these different wavelengths of light were most useful to plants. So he basically took a plate and he grew algae on that plate underwater. And then he introduced aerobic bacteria to the algae. Now, the aerobic bacteria could only live, or wanted to live, in the areas with the most oxygen. So what he found was the bacteria tended to congregate in areas where most of the oxygen was being produced. So as he basically exposed different wavelengths of light to different areas of the algae, he saw this congregation of bacteria, aerobic bacteria, in the reds and the oranges and in the violets and the blues. And he was able to then convert this into what's called an action spectrum, which basically shows what wavelengths of light do plants use the most. Well, they use the violets and the blues and the red. So this is basically sort of a direct application of what he saw in terms of where his bacteria were. Today we have spectrometers, and we have ways where we can actually shoot different wavelengths of light into plant pigments, and we can actually, within the different pigments, chlorophylls and carotenoids, we can see that they all absorb slightly different wavelengths of light. So chlorophyll A absorbs primarily over here in the violet range, but it also absorbs a lot of the orange and red wavelength. Chlorophyll B absorbs more in the blue wavelength, so it appears slightly different in color because of what's being absorbed and what's being reflected. And it absorbs again over here in the yellow and the red range. The carotenoids, these are pigments that you see in the fall when you get fall foliage. When the chlorophyll no longer produced, and you can see the reds and the oranges and the yellows in trees, at least up north on the East Coast where we have deciduous trees with a lot of fall foliage. You see all these pigments typically in autumn. And these carotenoids, again, they absorb more in the blue range, also some in the green range. They don't absorb very much in the yellow and the red because these are the colors that are being reflected. They're reflecting these back. But because we have all these different pigments absorbing different wavelengths of light, we're able to absorb much more of the visible light spectrum than if we just had one type of chlorophyll. So together they form these action spectrums where they can absorb more of the visible light spectrum because they all absorb slightly different wavelengths. Okay, so now let's talk about the light reaction. So the first step in photosynthesis is the light reaction, and you obviously have to have light. That's going to power this whole process. We're also going to start with a very low energy molecule, H2O. And we're going to take that molecule and we're going to excite those electrons. Remember, in cell respiration, we drained all the energy out of these electrons and then we just kind of stuck them onto oxygen and made water, this low energy waste product. So here we're going to take the energy from light and we're going to use it to excite these electrons, and then we're going to use those excited electrons to reduce carbon dioxide and make a high energy organic molecule, sort of cell respiration in reverse. Okay, so let's look at this in a little bit more detail. First, we have in these thylakoid membranes of the guarana, we have these photosystems, and it's basically chlorophyll molecules and carotenoids and these other pigments that absorb light. And as light comes down, photons of light, that is absorbed by them, and that energy is used to excite their electrons. Their electrons jump up into higher states. So they're absorbing that energy and they're getting their electrons excited. They're passing on those excited electrons to what's called a reaction center, right? Where there's one key pigment that's going to take all the energy in this reaction center and basically focus it on one electron which gets really excited. So when we do that, we're going to use that energy to split water and make oxygen, and also generate the molecule ATP, and the reduced molecule, we're going to create a reduced molecule called NADPH. Again, this sounds a lot like NADH in cell respiration, but there's a P in there, and you can just remember P for photosynthesis. This is the electron carrier involved in photosynthesis, not cell respiration. Okay, so let's look at this process in a little bit more detail in the light reaction. So here's a good analogy. We can think of these photons of light as like providing the energy, like this construction worker powering this electron up to a higher energy state. Okay, and this is in what's called photosystem II. Okay, this construction worker is going to pass down these electrons through this thing called an electron transport chain. Okay, that should sound familiar. That's the same exact process that occurs in cell respiration. And as this electron goes from high energy to low energy, that energy is going to be used to make the molecule ATP. Okay, exactly like cell respiration. So now that electron's been passed over to another area called photosystem I. We go from 2 to 1 because 1 was discovered first and then 2 was discovered, but really this process starts in the photosystem II and then goes to photosystem I. Anyway, we're now at photosystem I. We've got a low energy electron, but we're going to get more energy from the sun, and we're going to power that, and we're going to excite that electron up again. And we're going to use this now high energy electron to reduce the molecule NADP, it's not working very well, P, into a reduced high energy molecule called NADPH. Just like we made NADH from NAD in cell respiration, we're going to do the same thing here. We're going to make a reduced high energy molecule that's going to carry these high energy electrons into the Calvin cycle. Okay, so let's look at this in a little bit more detail. So here's photosystem II. Again, we're on these thylakoid membranes in the chloroplast. They're absorbing light, they're passing that energy around, and they're focusing all that energy onto one chlorophyll molecule, which excites those electrons. Those electrons are now in this very high energy state. And again, instead of just releasing that as waste heat, they're going to pass that excited electron down an electron transport chain and use that energy to make ATP. Okay, so this first step in photosystem II, we've excited an electron, and we've made ATP. Now, since we've lost an electron from this photosystem, we have to replace those electrons. So we're going to take water, we're going to strip off the low energy electrons, and we're going to strip off the hydrogen, and we're going to release the oxygen. So this first step, we've taken water, released the oxygen, we're going to keep the hydrogen ions, and we're going to keep the electrons because we need to keep exciting the electrons, and the hydrogens will be used in the next step here, which I'll explain. Okay, so now we're over at photosystem 2. We're going to take more light energy, and we're going to, again, excite chlorophyll, and then when this chlorophyll electrons get excited, they jump up to this higher energy state, and we're going to use those excited electrons to reduce this molecule NADP into NADPH, right? And here are some of the hydrogens that were stripped off the water over here that are going to be used to attach onto NADPH. So this is the basic scenario here. We're going to take light, we're going to use it to split water. We're going to those excited electrons are going to generate ATP in the first step, and then they're going to generate NADPH. And the ATP and the NADPH will be used in the Calvin cycle to make sugar from carbon dioxide. Okay, so again, this is very similar to what's going on in the mitochondria, right? The inner membrane of the mitochondria where we have electron transport chains. We have the exact same thing in these thylakoid membranes on the chloroplast. So what's happening with these excited electrons as they're being passed down to lower energy is that's going to then be used to pump hydrogen ions, in this case, not in the inner membrane space, that's what happened during cell respiration, but into these thylakoid membranes. So the thylakoid membranes are going to fill up with hydrogen ions, and then we can use that concentration gradient, all those hydrogen ions, to power this process through this enzyme, again, ATP synthase, which is going to make ATP, exactly like the mitochondria. So again, it's the same process of the electron transport chain being linked up with chemiosmosis. This process of hydrogen diffusing back out is called chemiosmosis, and that's generating ATP in the chloroplast just like it did in the mitochondria. This is what it looks like in more detail, and this should look a lot like that electron transport chain in the mitochondria, right? So we have, in this case, proteins passing electrons. We're stripping the hydrogens and electrons off of water, and we're exciting those electrons and passing them down. And we're, again, using that energy to pump hydrogen ions into the thylakoid membrane inside this thylakoid space. And then this concentration of hydrogen ions is going to flush out through this enzyme ATP synthase, which uses that concentration gradient to convert that sort of potential energy into the energy of ATP. Over here it's photosystem 1. We are taking more light. We're going to excite those electrons again, and we're going to use those excited electrons to power the production of ATPH, which is a reduced form of NADP into NADPH, which is then going to be involved in the Calvin cycle. So basically we've made the two ingredients that we're going to need to power the Calvin cycle, where we take carbon dioxide and fix it into a high energy organic molecule. Okay, and again, one other note in here, in photosystem 1, we also have a secondary process called cyclic electron flow. Notice the electron flow from photosystem 2 to photosystem 1 was non-cyclic. It went in one direction. We have to keep splitting water to provide more electrons. But we also have a secondary system here. We're taking excited electrons in photosystem 1 and sending them back down an electron transport chain to make more ATP. This is required because in order to make sugars, you need a little bit more ATP than you do NADPH. So we have to make a little bit extra ATP in photosystem 1. So this is called the cyclic flow of electrons in photosystem 1, which is going to give us a little bit more ATP for that process. Okay, so in the next video we'll talk about how we take the NADPH and the ATP from the light reactions, combine them with carbon dioxide in the Calvin cycle to make sugars. But that's it for this video.